For genetics, exam 3, part 2, I chose prompt B. First, we are going to talk about Nirenberg and Matei's experiment. Together they found that specific RNA sequences code for specific amino acids. They were able to identify 3 codons of the total 64 that are eventually identified. So how did the two of them do it? Well, their recipe for success started with 2 simple ingredients. They needed 1 poly-U, or a long strand of U's, and 20 different radioactive labeled amino acids. Then they got started. They combined each different amino acid with a strand of the poly-U in separate tubes. Then they had to look for the tube that showed the newly synthesized protein that was also radioactively labeled. This would show that the amino acid chain that was radioactively labeled was part of the protein formed. The poly-U formed a chain with the amino acid phenylalanine. This meant that the UUU coded for phenylalanine. They had figured out one code. Then they tried with 3 more RNA sequences. For each one, they repeated the same steps. However, they replaced the UUU with either AAA, CCC, or GGG. They found that AAA coded for lysine, CCC coded for proline, and they were unable to figure out the code for GGG. Next, Nirenberg paired up with another scientist, Letter. Together, they identified the code for the other 61 codons that Nirenberg and Matei did not identify. Their recipe for success had some similar ingredients but a slightly different approach. They used trinucleotides. In this first example, they used GUU, radioactive labeled amino acids, ribosomes, tRNA, and a nitrocellulose filter. To start, they took the trinucleotide GUU and added it to a mixture of ribosomes and tRNA. Then they put that mixture through the nitrocellulose filter. The last step was to look for which specific tRNA binds to the ribosome and mRNA complex. This should all be stuck on the filter as everything else that didn't code filtered through. They found the GUU and ribosome complex bound with tRNA for valine stuck in the filter. This showed that the mRNA sequence GUU coded for valine. Then they repeated these steps, substituting all other trinucleotides for the original GUU to determine what amino acid they code for. This was significant because it provided us with the full coding dictionary that we use to transcribe amino acid sequences from mRNA to this day. Thanks for watching. That's the end of this presentation.